Okay, so we're going to have a look then at the factors that affect the rate of gene expression. So there's three main factors that affect the rate of gene expression. You can either talk about transcription or epigenetics or RNA interference. So transcription and translation are the processes by which you start with your DNA. So this is inside the nucleus. You will make a copy of one strand using RNA polymerase. That's called your pre-mRNA. Then it undergoes splicing. So you remove, splicing is where you remove the introns and you're left with the exons which get put together by the enzymes, and that is your mRNA. Now the mRNA will then leave the nucleus and be translated using a ribosome. That's because the mRNA is single-stranded and the tRNA anticodons are complementary to the codon in the mRNA. They line up according to complementary base pairing, and as they do that, they cause the amino acids that they carry in a very specific manner. So only one type of tRNA will carry one type of amino acid, so that the code that they carry, so the anticodon that the tRNA carries, corresponds specifically to one particular type of amino acid. They will be held close enough together by the ribosome so that a peptide bond forms between the amino acids, creating a primary structure protein, which is the sequence of amino acids. So what we're talking about in the regulation of gene expression is something that will either interfere with transcription, so that's the copying by the enzyme RNA polymerase. So it could be if something affects how easy it is for that RNA polymerase to get onto the DNA to make a copy to make the mRNA, then that will affect the rate of transcription. So the easier it is, the faster you'll get to your transcription, so the more mRNA you'll make, so therefore the more protein you'll make. The harder it is for RNA polymerase to get on, the more difficult it will be to make mRNA, therefore there will be less mRNA and less protein. Then the other way of interfering with gene expression is something to do with the translation step. So if you put anything or bind anything and stop that ribosome from binding to that mRNA, then you will stop translation and you will not get the protein. So, first things first, transcription factors. Transcription factors are exactly what the name suggests. They are factors that affect how well transcription can occur. So if your aim is to unwind the gene in the unwind the DNA in the region of a gene and allow your RNA polymerase to bind, it will bind at the site called a promoter. So you can have molecules, transcription factors, that can either act as an activator, so for example they can bind to that promoter and make it easier to trap the RNA polymerase. So activators do what they say, they activate, make it easier for the RNA polymerase to bind, therefore you will get your transcription, so you'll get more mRNA. So activators cause more mRNA to occur. On the other hand, you have repressors, which are basically molecules which will bind to the DNA, and when they bind to the DNA, they will, at the promoter, they will prevent the polymerase from being able to bind. And if the polymerase cannot bind, the RNA polymerase cannot bind, it cannot make mRNA, so you cannot get a protein. So those are ways of regulating. So transcription factors are useful because if you have, for example, a use for that protein in the cell, if you want to make lots of that protein because you need it, for example, if it's an enzyme, say for example an enzyme that breaks down lactose, then what you would want is lots and lots of the enzyme beta-galactosidase to break down that lactose into the glucose so that you can respire it. However, if there was no lactose in the cell, what is the point of making an enzyme that has no substrate to work on? And in that case, what tends to happen is you will have a repressor that binds to the promoter region for that particular enzyme's gene and stays there until you've got lots of the substrate. So in the case of the lac operon, you find that the lactose substrate can bind to the repressor, the lac repressor, and that will cause it to be removed. So when lactose is present in the cell, it binds to the repressor, that causes it to not be able to bind to the promoter, so therefore your RNA polymerase can bind to the promoter region. You do get transcription, therefore you do get mRNA, so you do get the enzyme beta-galactosidase needed to break down your lactose. So it's a good way of regulating gene expression, because when you need a gene to be transcribed, it will be, and when you don't, it won't be. Okay. So the second way of controlling gene expression is through epigenetics. Epigenetics is where you can have molecules or epigenetic marks, for example methyl groups or acetyl groups are two examples of epigenetic marks that can bind to either the DNA, so methyl groups can be found between C and G bases in the DNA, and the methyl groups will attach in these regions anywhere where there's a C and a G, and because those are extra groups on the DNA, they make it very difficult for your RNA polymerase to get access to the promoter regions, so therefore you decrease your transcription. So we can say, for alliteration purposes, that methyl groups mute the transcription, so they mute the expression of the gene. So methylation mutes the expression. Acetyl groups, they actually bind to, if we remember, chromatin is bound around histone proteins. And the more tightly it's bound around those histone proteins, the more difficult it is to get RNA polymerase on. So what acetyl groups do, the acetyl groups bind to the DNA and help to keep the DNA loose around the histone proteins. And because of that, it makes it easier for RNA polymerase to bind where you have acetyl groups. So we say that acetylation accelerates gene expression. So if we have acetyl groups on a gene that we want, then we know that we'll be more likely to get RNA polymerase binding to the promoter, so more machinery for the transcription. If you get more transcription, you get more mRNA, and you will get more of that protein. In contrast, methylation mutes or silences or turns off gene expression. It makes it more difficult for the RNA to bind, and therefore less transcription, less protein synthesis. So methylation mutes and acetylation accelerates gene transcription. Okay. These are different ways that you could actually use methylation and acetylation, which we'll come on to later when we talk about cancer treatments, as a way of modifying gene expression to help prevent tumours for cancer treatment. Okay, so the last one is interference RNA. So this isn't to do with transcription anymore, this is to do with translation. And we know that translation occurs on mRNA that is single-stranded. So once the mRNA has left the nucleus, it is able to bind to a ribosome, and the ribosome can read the codons because they are exposed, because it is a single strand. The codons are read because there are complementary base sequences in the anticodons of your tRNA, and you make your polypeptide, etc. Now, in the case of interference RNA, there are two types, and you need to know the names of those, which is microRNA and small interfering RNA. The only real difference that you need to be aware of is that microRNA may not be a perfect fit. So the base sequence, complementary base sequence, might not be 100%, but it still works because some of the bases are complementary. So this means that microRNA could potentially bind to mRNA for a number of different base sequences from a number of different genes. So this makes microRNA more generalized. And once it is bound, it means that the ribosome cannot attach, because it will not attach to double-stranded RNA. So that means you do not get translation. Small interfering RNA, in contrast to microRNA then, is a perfect fit. So the sequence of the small interfering RNA will be exactly complementary to the mRNA that it is going to cause to be degraded. And what happens in both cases is there is a complex called RISC. And RISC is RNA-induced silencing complex. And what that's going to do is it's a setup of proteins and your microRNA or your small interfering RNA, and when it binds to the RNA, the mRNA that's going to be translated, in the case of this example, your protein complex, which is the RISC complex, along with the small interfering RNA, will cause RNA hydrolase, which is an enzyme able to break down phosphodiester bonds. So hydrolyzes phosphodiester bonds. It will cause the mRNA to be chopped up. And if you don't have any mRNA, then you definitely can't do translation and you won't get a protein. So the main things that you need to understand for the RISC and interference RNA is that there are two types of interfering RNA, microRNA and small interfering RNA. The main difference between them is just whether or not they bind completely and perfectly by complementary base pairing, or there's a slight difference in the base sequence. So that means that the microRNA could bind to a number of different mRNAs, whereas the short interfering RNA probably only binds to one specific gene mRNA. So one mRNA made from one specific gene. That's useful in the cell because it means that if you have in the DNA one promoter, it could make the mRNA for the gene that you want, and it will also make a section of small interfering RNA. That way, once you've made enough of the protein that you're interested in, your small interfering RNA would come along and stop you making any more. And that helps you to prevent you from making too much of that protein. Maybe it's too high energy requiring, or maybe there's only a limited amount of protein that you actually need. And there's no point wasting the resources of the cell, so it's a good way of putting a stopper on the production line when you don't need it to keep going on and on and on. MicroRNA is good if, for example, you had RNA coming in which is single-stranded from, for example, a virus. So if a virus is trying to infect a cell, it will insert its short strand of RNA, its RNA sequence, because remember, not all viruses have DNA, some of them use RNA, and it's then reverse transcriptase that binds to it, makes it into DNA, gets into your host cell's DNA, and then that host cell's DNA can make the viral proteins. Well, if you can get this microRNA to bind to the, for example, HIV virus RNA before it gets a chance to make a copy of itself and gets into your host cell DNA, then it can be degraded, and that means that you cannot allow that virus to replicate inside yourself, and so your cell has a way of combating viruses. And so there are a number of plant and animal cells that are able to make that to combat that phenomenon. Okay. The last thing is that you appreciate that both of these setups require the RNA-induced silencing complex. So this is not a random setup. This isn't something that just happens at random with any piece of mRNA. You first of all have to have a complementary sequence of mRNA, and you have to have a sequence of proteins that are also able to combine, to come together, to signify that this is the mRNA that's either going to get chopped up, and if it's chopped up you can't do translation, or maybe it will be stored for later, which can sometimes happen in this case, or it just gets covered with the RISC and the microRNA together. And if you double strand your mRNA, it cannot be read. And if it cannot be read, you cannot get translation, you cannot get proteins. Okay, any questions on any of that? Okay.